Preparation model paper 21. I'm N. Shankar Reddy, PhD plant pathology. The activity of antagonistic fungi is more in dash soil. So sick soil, suppressive soil, no suppressive soil, field soil. So they have asked antagonistic activity, which means the biocontrol activity is more in which soil? Suppressive soil. So where the biocontrol activity is more. So the next one is a specific region of viral antigen that induce and interfere with the specific antibodies are termed as, I repeat, specific region of viral antigens that induce or interrupt with the specific antibodies are termed as tier, C terminus, N terminus and epitopes. Generally it is called as the epitope. So epitope is generally a viral antigen that induces the specific antibodies, or the production of a specific antibodies is known as a epitope. Okay. Tobacco streak virus is a type member of the genus. They are simply asking a tobacco streak mosaic virus belonging to the genus. It is belonging to the genus ICAR virus. So the next one is Dahlia mosaic virus is an example of genome of Dahlia mosaic virus. They have asking single stranded RNA, single stranded DNA, double stranded RNA, double stranded DNA. So Dahlia mosaic virus contains a genome of double stranded DNA. Along with this Dahlia mosaic virus, one more virus like cauliflower mosaic virus also contains a double stranded DNA as a genome, right? So next question, National Pest Risk Analysis Center in India has been established in. So where they have established the National Pest Risk Analysis Center in India. So it is established in actually Mumbai. First A-virulent gene AVR. AVR is the first A-virulent gene, especially fungal A-virulent gene. If I want to tell you an exact, AVR6 was isolated from a Cladosporium fulvum. So tomato leaf mold fungus, Cladosporium fulvum. So that was isolated by, who isolated this first fungal A-virulent gene AVR6 or CF9 isolated from a Cladosporium fulvum. DeWitt, Clussig, Royals and Briggs. So the first fungal A-virulent gene was isolated by PJGM DeWitt in 1991. Next one, Moco wilt of banana is caused by, they are asking caused by Irvinia amylovora, Ralstonia, Fusarium oxysporum, Pseudomonas solanacearum. We know very well Moco wilt of banana is caused by Ralstonia solanacearum. Tyndallization is performed at, what is the temperature of tyndallization? So tyndallization is a specialized technique which was developed by John Tyndall for the killing of bacterial endospores. Bacterial endospore cannot kill at a normal temperature or normal heat. There is a specialized technique for the killing of endospores called tyndallization. Tyndallization is the technique recommended for the killing of endospores. If we see the bacterial genuses like Bacillus and Clostridium generally produces this endospores or a heat resistant spore. So why endospores are that much heat resistance? Because it contains an acid called DPA, deep dipiconic acid, where the calcium ions are more at that in DPA. So due to that it is resistant to heat. So the temperature is 100 to 20 degrees and 100 degrees Celsius for 20 minutes for 2 days, 123 days, 122 days, 120, 122 days. Generally 100 degrees Celsius for 20 minutes for 3 consecutive days. It is also called as a discontinuous heat of sterilization, because we are heating or sterilizing in a discontinuous way. So in first day we are heating at 20, and the next day again 20, and the next day again 20. So it is a discontinuous method of heating, right? So it is the best example, and sometimes they may ask to give an example of discontinuity of sterilization, like tyndallization is the best example. Pasteurization temperature is a 62.8, right? So but tyndallization is 100 degrees Celsius for 20 minutes and 3 consecutive days. Next one is a commonly used percentage of sodium chloride as a surface disinfectant. 0.1%, 0.5, 1% and 3%. Generally if it is a mercury chloride we recommend at the rate of 0.1%. Since it is a sodium hypochloride we recommend at the rate of 1% concentration. Downy mildew is caused by the members of the family. So which of the following member is responsible for the downy mildew fungi? Pitheaceae, Albuginaceae, Peronosporaceae, Monilinaceae. Actually, before it is downy mildew are classified under Pitheaceae. Now it is actually classified under Peronosporaceae. Okay, all downy mildew fungi now it's classified under Peronosporaceae. Before it is under Pitheaceae. Okay, wound hormone. Which of the following is a wound hormone? Pisatin, osmotin, tramatin, and auxin. So tramatin is the wound hormone. Okay. Beta 1, 3 gluconases is a PR protein 2, PR protein 3, PR protein 4, PR protein 5. Generally, beta 1, 3 glucanase is a PR protein 2. Okay, PR protein 1 is chitinases, PR protein 2 is beta 1, 3 glucanases, 5 is the tomato like protein, proteins, such kind of things will be there. So far, there are 17 kinds of PR proteins have been identified. Chitinases, beta 1, 3 glucanases, tomato like proteins. These are a couple of PR proteins. Coconut root wilt is caused by virus, phytoplasma, spiroplasma, and viride. Generally, this coconut root wilt is caused by phytoplasmal organism, right? Parthenogenicity, parthenogenetically developed zygospores are called as parthenozygospores, azygospores, conidia, and sporangia. So the development of zygospore through the parthenogenetic process or the parthenogenetically developing zygospore is known as azygospores. So zygospore is a sexual diploid resting spore, right? We know very well. That is produced by Zygomagota group of fungi. So beet necrotic yellow vein virus is transmitted by Polymyxa graminis, Polymyxa betae, Olpidium betae. So all the above. So among the given options, this Polymyxa betae is the most suitable answer for this beet necrotic yellow vein virus. Sex pilus or conjugation pilus is formed by the bacteria during. So during which recombination process this sex pilus or conjugation pilus can be observed? Through the process of conjugation only. The sex pilus is also known as conjugation pilus. Among the three kinds of sexual recombination, there is only one recombination where bacteria is directly connected by using a tube called sex pilus or conjugation pilus. That is called conjugation. So this technique was developed, this kind of recombination was discovered by Letterberg and Tatum in 1946. ELISA is made up of, you know very well what is ELISA, enzyme linked immunosorbent assay, right? That was developed by Adam and Clark in 77, right? So they're asking the plate is made up of, ELISA plate is generally made up of a nitrocellulose or nitrocellulose membrane we used to call, right? So Huan Lang Ping, we can also call it as a citrus greening disease. It is a very important disease which is caused by fastidious vascular bacteria, Candidatus liberibacter, Asiaticus, Americanus. There are a couple of species are there, but generally we used to study Asiaticus in Asia, Americanus in America. Okay, so when it comes to our Asian countries like China, India, the citrus greening is generally caused by Candidatus liberibacter asiaticus, which is a fastidious vascular bacteria and is transmitted by, they're asking, it is transmitted by insect vector called psyllids. It is transmitted by insect vector called psyllids, Diaphorina citri, I think the scientific name. So which of the following mode of transmission is not associated with the virides? I repeat, not associated with virides. Vector, grafting, mechanical, cultural. See, if you see, majority of the plant virus diseases are transmitted by vectors, insect vectors. But there is no viride have been reported so far that is transmitted by insect vector. Since they are asked not associated, it is a viride. So grafting kind, mechanical transmission and cultural practices are the most suitable or most normally reported kind of transmissions in virides, and there is no vector transmission have been reported. So far there are 40 virides have been discovered. All the 40 virides are known to be transmitted by mechanical or maybe grafting or maybe cultural. Okay, no vectors have been reported. Okay, virus free certification program was developed in, so which of the following crop where a virus free certification program was developed? Cucumber, potato, tomato, brinjal. So virus free certification program was actually developed for potato, because if you see all the crops, there is only one crop which is infected by more than 75 or 76 type of viruses, that is potato. In potato itself, more than 70 plus type of viruses have been reported. It's a very important to develop a virus free plantation for culture, right? So this virus free certification was actually primarily developed for potato. Various potato diseases are there, right? Bimodal transmission seen in the case of which of the following viral disease where bimodal transmission, like bimodal transmission in the sense to be simple, transmitted by means of two means, like one is like insect vectors are the first one is, second one is by means of other. So if it is transmitting by means of two modes or two different modes, it is known as bimodal transmission. So Gemini virus, Calimovirus, Bromovirus, Topomovirus, as far of my knowledge, it is best known for Calimovirus, like cauliflower mosaic virus. So the cauliflower mosaic virus is known to be transmitted by two measures, two means. The first one is aphids, the second one is mechanical transmission or sap transmission. So this bimodal transmission means two means of transmission. Best example is the cauliflower mosaic virus. I think cauliflower mosaic virus comes under this Calimo virus or Calimoviridae, I think. Simply spraying of water can reduce this. Which of the following disease? At the initial stages, if we spray water itself, we can manage the disease. Bacterial disease, powdery mildews, alternate leaf spot, downy mildews. Best example is powdery mildew disease in the initial stages. So one of my professor also used to say this, at the initial stages, if the cells are not colonized or not penetrated or not colonized with this leaf, simply spraying of water is more than enough. On the next stage, we can go with 1% milk solution. If it is too much or too heavy, or the heavy infestation will be there, then you can go for powder mildew kind of fungicide like tridomorph. Such kind of fungicide can be recommended. So but simply spraying of water at initial levels can be greatly helpful to just wash out the spores which are present on the surface of the leaf. So this powdery mildew kind of disease at the initial stages, spray the water at the initial stages, not at the severe stages. Okay, so which of the following viruses are not commonly used as a vehicle or vector used for the genetic transmission? So CMV, TMV, PVX, cauliflower mosaic virus. So this cauliflower mosaic virus is the most commonly used vehicle in transmission, or used as a vector for transmission of various kinds of plasmids by using through this genetic engineering, in the genetic engineering process. Spores that are formed by the budding are known as, simply budding spores are known as blastospores. Budding spores are known as blastospores, or spores that are developed through the process of budding is also known as blastospores only. Who established that nucleic acid alone is the infectious material, which means nucleic acid is alone is enough to cause the infection, or nucleic acid is the actual original culprit in the causing of disease. Before, it is used to believe that, for example, even W.M. Stanley also told that protein coat is the infectious material, but it was wrong. Later it was came into conclusion that nucleic acid is the infectious material, where protein coat only protect the nucleic acid, and protein coat has no role in disease spread, disease transmission, and disease causing. Only nucleic acid causes the disease. That was given by W.M. Stanley, Geyer and Scram, Bowden and Pyre, Gibson and Harrison. So Geyer and Scram in 1956, this question was asked in NET 2 times. Geyer and Scram in 1956, they told that nucleic acid is the infectious material of plant virus. If you see here, Bowden and Pyre, they also have done the greatest contributions. Like they called a virus as a nucleoprotein, which means that's a combination of a nucleic acid and a protein. It's called a nucleoprotein. So they gave this nucleoprotein concept in 1936. In fact, instead of W.M. Stanley and other Nobel laureate, they might have got the Nobel Prize, but unfortunately they missed the Nobel Prize. That's okay, no problem. But still, they have done the greatest contributions to the field of virology, undoubtedly. And so the answer is Geyer and Scram, who they have established a concept that nucleic acid alone serves as the infectious material of plant viruses. Next one is example of a mycorrhizal fungus. Which of the following is a mycorrhizal fungus? Penicillium, Pezizia, Ericife. So Ericife generally causes powdery mildew. This is a mold Penicillium. Pezizia, it's a kind of mushrooms, right? So generally glomus species, various kinds of glomus species are there. Glomus mosae, other kinds of species also there. They are actually the AM fungi, arbuscular mycorrhiza fungi, or vesicular arbuscular mycorrhiza fungi. Now its name changed to AM fungi instead of vesicular arbuscular. Now it's called as AMF, arbuscular mycorrhiza fungi. Okay. So fungi belonging to the which order are obligate parasites? First of all, we must understand what is obligate parasite. Like they simply required, strictly required a living host for their survival. Agaricales, mushrooms, taphrinales, peach leaf called plasmodioporales, like clubroot of cabbage. So they all are not strictly obligate parasites. When it comes to histologenates, these histologenates is a very strictly when compared to others. Like if you see this plasmodioporal, also like slimy molds we can found in water. But these histologenates, especially rust kind of fungi, They're strictly obligate parasite. They need a living host for the survival. Okay, so the answer is the Eustulaginales. Cross protection is effective in controlling which of the following virus infection? So Badna virus, Tobacco virus, Madari virus and Clastrovirus. So which of the following is this cross protection is very effective? So this Clastrovirus, for example, if this citrus virus comes under this group of Clastrovirus only, it's a very important virus where stem pitting is the most significant character we can observe in this citrus virus. Managing this citrus virus by using antibiotics and other varieties, resistant varieties, found a little bit difficult, but there is a technique called cross protection where we can effectively control this disease. So plant viruses database wide, actually I couldn't able to recall this full terminology, but I have came across this wide database somewhere else. Generally this database is actually represents plant viruses. Okay, this database is located at USA, Japan, UK and Australia. Actually it is located at Australia. Okay, it was maintaining by Australian people. Wide transfer. Okay, the next question itself is a wide transfer, right? So virus identification data exchange, virus identification data analysis, virus identification data analysis, virus indication data exchange. I believe it's a virus identification data exchange, probably. I'm not sure. I suggest you to please refer what is the true expansion of wide. I'm little bit doubted, but probably it is might be the virus identification data exchange, probably. Okay, acid fast staining is recommended or used for Mycobacterium, Xanthomonas, Cyanobacteria and bacteriophages. This acid fast staining is recommended for a bacteria called Mycobacterium tuberculosis, and this bacteria is also the best example for both gram positive and gram negative bacteria. Which of the following sprayer is used for high volume spraying? So among the given sprayers, this plunger pump sprayer is seems to be the high volume spraying. Of course rocker spray is also, but compared to the rocker sprayer, this plunger pump sprayer is the high volume sprayer. Pyrimidine fungicides are effective against. So this pyrimidine group of fungicides are effective against this powdery mildew group of fungicides. Fungicide recommended for post harvest is dipping. So which of the following fungicide is a post harvest fungicide? The best post harvest fungicide in the given options is thiabendazole. So this thiabendazole is recommended by the best post harvest dipping fungicide. Fungal nematicide. So which of the following is a fungal nematicides? Alternaria casei, Colloidum glospirides, Arthobotrytis robusta, all the above. So this best example is Arthobotrytis robusta. In mycology also, this fungal traps are fungal mycelium actually traps the nematode. That's why it's called as fungal traps. This mycelium actually traps the nematode and kills the nematode. That's why it's called as a fungal nematicide. So Arthobotrytis robusta. Fluid mosaic model is related to the structure of. So the fluid mosaic model is actually related to the structure of plasma membrane. Spongy tissue of mango is a bacterial disease, fungal disease, viral disease, physiological disorder. So spongy tissue of mango is a physiological disorder. Which of the following is not a Basidiomycota group of fungi? Among the given options, this Chaetomium globosum is a biocontrol agent. It is not actually belonging to this Basidiomycetes. Perfect stage of Rhizoctonia solani. So Rhizoctonia solani, which generally causes sheath blight, right? So spelling also a little bit wrong. Please make it correct. Rhizoctonia, not Rosoctonia. It's a Rhizoctonia. So the teleomorphic stage of or perfect stage or teleomorphic stage or sexual stage of Rhizoctonia solani is Thanatiphorus cucumaris. Cleistothecia is produced by, you must understand what is Cleistothecia. Like it's a completely closed ascocarp. Like if you see in Ascomycota, there are 4 kinds of sexual fruiting bodies we can observe. Cleistothecium, Apothecium, Perithecium, Necrodaceae. Cleistothecium means it's a completely closed ascocarp where we mainly observed in Ascomycota group of fungi. In Zygomycota and Basidiomycota don't produce any such kind of ascocarps, right? So pinwheel inclusion bodies are produced by. So TMV, Rio viruses, cauliflower mosaic viruses and Poti viruses. Actually Rio viruses produces generally a pinwheel kind of inclusion bodies. ISTA stands for International Seed Testing Agency. sorry, International Seed Testing Association. It's not agency, International Seed Testing Association. It is a ISTA. Which of the following fungus does not produces juice spores? I repeat, juice spores. Pythium produce juice spores. Albo produce juice spores. Uncinella nicator. It is actually powdery mildew, right? So powdery mildew won't produce any juice spores. It produces conidia, right? Plasmopara, it is also produces juice spores. All the given fungi, this Uncinella cannot produce or does not produce juice spores. Okay. Which of the following is not a resistant gene for fungal pathogens? SR23, probably the rust. CF9, Cladosporium fulvum, tomato leaf mold, HM1, first resistant gene. This Xanthomonas 21 is a resistant gene for BLB, rice bacterial leaf blight, right? Since they've asked not a fungal resistant gene, so the answer is Xanthomonas 21. Type 3 effector system mostly present in. So there are, if you see the bacteria, around 6 to 7 types of secretion systems are the type 1, majority of the plant pathogenic bacteria. Type 3, toxins producing kind of the secretion system. Type 4, agrobacterium tuum fascium. Such kind of secretion systems are there in bacteria, right? So they are asking mostly present in plant growth promoting bacteria, plant symbiotic bacteria, nitrogen fixing bacteria. So generally type 3 secretion system are mostly present in plant pathogenic bacteria. The two toxin producing kind of virulent disease causing kind of plant pathogenic bacteria must having this type 3 secretion system. So management of plant viral disease is possible through the management of plant viral disease mainly we can possible through pathogen derived resistance, RNA silencing resistance, all the means of above given options we can achieve the managing the plant viral diseases. The first fungal avirulent gene was isolated from Elementosporium carborum, Elementosporium victoriae, Cladosporium fulvum, Pyoidother infestation. Just now we discussed Cladosporium fulvum, right? That is a tomato leaf mold fungi that is known to be the first fungal avirulent gene that was isolated by PJGM David 1991. Planned geographical distribution of major genes for a race specific resistant of the pathogen is known as, it is generally known as gene deployment, where major genes are distributed and race specific resistance, that is in the major geographical areas, is generally known as gene deployment. Mode of action of tab toxin. Tab toxin is a Pseudomonas syringae pathover tabaci. The mode of action is ornithyl carbamyl transferase, that is the general mode of action. The citrus canker lesions are characterized by the over deployment of, I repeat, citrus canker lesions are characterized by the over development, it's not a deployment, over development of parenchymatous tissues, epidermal tissues, ectodermal tissues, and probably parenchymatous tissues, I'm not sure. Just please take a look into a form of canker in a citrus. A form of canker in a citrus is caused by, I don't know what is the meaning of this question. A form of canker in citrus. Anyway, let's see. Xanthomonas campestris pathover citrus, not citrus canker. Xanthomonas axonopodus pathover citrus. Okay, it is caused by citrus canker. Xanthomonas axonopodus pathover aurantifolia. It is caused canker and some other recipes probably. Among the given options, this Xanthomonas axonopodus pathover citrus seems to the reliable answer, but it's a little bit doubtful what is the form canker of citrus. But anyway, generally citrus canker is caused by, among the given options, Xanthomonas citrus pathover citrus, and sometimes you can also observe the other options like Xanthomonas smithi, PV smithi. Xanthomonas PV citrus kind of options also might be there, but generally this option we can expect more. Xanthomonas axonopodus pathover citrus. The little dilution effect is found in Xanthomonas and bacteria, Pseudomonas and bacteria, RSD, probably Xanthomonas and bacteria, I think. So lethal dilution effect on the general count in Xanthomonas and the other type of bacteria probably. So which is known as a true canker among the given options, which is known as a true canker? Mango canker, citrus canker, butternut canker, tomato canker. Among the given options, the citrus canker is known as the true canker. Grassy shoot disease was first noticed in which state of India? In the state of Maharashtra, this grassy shoot was first observed. Lead blight of potato was first introduced in India. Which place? Ooty, Rajasthan, Nilgiris, and Himalaya. It is believed that Nilgiri Hills is the first area where this lead blight of potato was introduced in India. Angular leaf spot of cotton was the first reported from which country? USA, China, India, and Japan. I think angular leaf spot of cotton, that is bacterial wilt of cotton, was first reported from USA. It was reported by, I think, Atkinson in 1892, probably. Atkinson in 1892 only discovered this. First identified this bacterial blight of cotton, I think. USA is definitely, but generally, definitely USA only. But Atkinson was the first person, I think, who first discovered this angular leaf spot of cotton. Which Pseudomonas bacteria act as a plant pathogen? Pseudomonas aeruginosa, Serenge, Putida, Pseudomonas fluorescens. Among the given options, the Pseudomonas fluorescens recently emerged as human pathogen also, like it causes breathing kind of infections. Of course, we know wherever it's a Pseudomonas fluorescens, we are using from decades onwards, it is a biocontrol agent. But in recent genomic analysis reveals that it is also a plant pathogen. It leads to respiratory diseases, severe respiratory disease. Okay, the black 47 is related to. Black 47 is related to Irish famine. I will tell you what is this black 47. Black 47 related to which crop? Potato crop. Irish famine, late blight of potato. I will tell you this black 47. Generally, in 1945, late blight of potato occurs, right? In 1947, the condition of the people in the Irish is very, we cannot expect or imagine. Nobody have food, and like everyone is struggling for food. Like many people are dying just because of food starvation. And here there is a movie called Black 47, which was actually released in 2018. In 1845 and 47, late blight of potato occurs. In the same time, there is a war going on in England and other countries as well. It will be very difficult for the people to survive without food. And that movie Black 47, that shows the that show you the exact scenario how actually the people suffered that time. But majority of the movie concentrated on war only, but they have also showed the indirect effect of this late blight of potato as well. But the movie is fantastic and a masterpiece. I suggest you, if time permits, please go and take a look into this movie. It's a fantastic movie, there is no doubt about it. This movie mainly concentrated on the serious effects of late blight of potato along with the war as well. Okay, the anti-tumor chemical clavation is present in the fungus. So which fungus it contains clavation is the anti-tumor properties. So there is a fungi called puffball fungi, clavaria. So okay, it contains a compound called clavacin, which is anti-tumor properties. Schizosaccharomyces pombe is used as a production of citrulline, model organism, and production of proteins from kerosene and diesel oil. So it is generally used for all the purposes. Among the given options, it is used for all the purposes. Sacred mushroom. Which mushroom is called as a sacred mushroom? Armillaria, psilocybe, agaricus coprinus. Psilocybe mexicana and Psilocybe semilantia generally considered as a sacred mushrooms or sacred fungi. Liberty caps are found in. So liberty caps are found in which of the following? Again, same Psilocybe mexicana and Psilocybe is only. Industrial penicillin is produced from Penicillium notatum, Penicillium chrysogenum, Penicillium griseofulvin. Generally, from all the fungi, the penicillin is producing, but at present, Penicillium chrysogenum is the fungi where everyone are recommending, because when compared to the other fungi, it will produce 100 times more yield than the other Penicillium species. Okay, now Penicillium is not using it. Almost become all the bacteria become completely resistant to this Penicillium. It's almost outdated. So the mixing kind of antibiotics now we are using, like streptomycin sulfate. Even if you see in human doses also, they are using a combination, cyclohexamide are like a combinations, right? So the penicillin is almost outdated. For the namesake and for studying the stuff, we can tell this penicillin is the first antibiotic to be discovered by Alexander Fleming 1928, and all those things we can say. So the answer is all the above. Okay, wilt of pea is caused by Xanthomonas virae, Fusarium vodum generally causes wilt of pea. Which of the following disease is called as a ring disease? Wilt of potato, citrus canker, black rot of cabbage, and angular leaf spot of cotton. Generally, wilt of potato, where we can see some kind of rings. That's why it's called as the ring disease, nothing else. And the next one is use of organic mercurial fungicide as a seed treatment for the wheat smut disease was the first recommended by Ream. Ream et al. Was the first recommended this mercurial fungicide for the wheat smut disease. Suitable medium for Ralstonia solanacearum. So Ralstonia solanacearum generally we can grow in a medium also, but the most suitable medium is the tetrazolium chloride medium. Literally the word penicillium means. So generally the meaning of penicillium is a brush like, I would say artist brush like. The word aspergillus means a toilet brush. It look like a toilet brush, but penicillium look like a artist brush or brush like. Okay. Assessment of chooses for foliar diseases like person disease instance, both A and B. None of the above. Assessment chooses the okay. For foliar disease, we can recommend a percent disease index only. Okay. Generally, this percent disease index, percent disease incidence, all generally recommended for the to assess the severity of the diseases. But for foliar disease, we can recommend percent disease index. Assessment chooses for a systemic disease. Obviously, we can go with the other option. This is percent disease incidence. Specific medium recommended for Phytophthora. There is a specific medium called V8 juice medium. V8 juice medium is the specific medium that we can recommend for this Phytophthora. Beef extract and peptone are used for culturing of fungal pathogens, bacterial pathogens, viral pathogens, all the above. Generally, this beef extract is the major ingredient for the culturing of bacterial plant pathogens, right? Fungi can also separate it in a culture media from bacteria by adding 1 to 2 drops of 25% solution of DAS that inhibits the growth of the bacteria. So very simple. If you add lactic acid, we can just inhibit the growth of the bacteria. Okay. Profuse upward branching of twigs are the symptoms of wilt, witch's broom, leaf spots, and the galls. Generally, witch's brooms where we can see the profuse upward branching. Flagellated protozoans belonging to the genus Phytomonas. Flagellated protozoans or flagellated Phytomonas as we can also call it. Test used for the detection of fungi. So, paper disease ke blotter say poison food technique and dual culture. What they have asked test used for detection of fungi. Okay. For the detection of fungi, we can recommend blotter paper. Okay. This is poison food technique and dual culture bioefficacy studies, right? So film forming compound, which are the following is a film forming compound which actually form film. Like a transparent film on it. Quinols, sulfur fungicides, anti-transparents, and dithiocarbamates. I don't know, probably anti-transparents forms that the name itself indicates anti-transparents, but I'm not sure about it. Please just take a look into. Spores formed by the fragmentation. So spores formed by the fragmentation is known as arthrospores only. Arthrospores are also known as idea, I think. Yes. Examples of meiospore, which are the following: oospore, geospore, ascospore. All are meiospores only. Like sexual spores are also known as meiospore. Okay. Thick-walled spores produced by the thickening of mycelium. Arthrospore, oidea, chlamydospores, and zygospore. Answer is chlamydospores only. What they have asked, thick-walled spores produced by the thickening of mycelium. So all the mycelium thickens and from a resting spores called chlamydospores. The same thing also can be observed in sclerotium as well, right? Tinsel flagellum consists of long rachis with lateral-like projections are called as mastoconiums, flimmers, both A and B, none of the above. So tinsel flagellum that consists of long rachis with like projections are called as both A and B. Answer: mastoconiums are otherwise called as flimmers. Okay. Trade name of tetracycline: streptocycline, griseofulvin, blastocycline, all the above. So the trade name of tetracycline is streptocycline. Protomyces are cruciform type divisions can be observed in. So protomyces are cruciform type of divisions mainly can be observed in Plasmodium mycota. That is a special features like observing of presence of Plasmodium, and cruciform type of divisions is the special characteristic feature of Plasmodium. Okay. During ascus development, karyogamy occurs in ascogenous hyphae, ascus mother cell, both A and B, and ascus generally. The ascus development only takes place in ascus mother cell. Okay, ascus development only takes place in ascus mother cell. The drug cyclosporin used in transplant surgery derived from penicillin as per Gilles, Toliposporium, all the above. Probably Toliposporium, I think the name itself, Toliposporium, Cladosporium, because in Penicillium and Aspergillus there is no such kind of, I never heard there is no such kind of drugs are isolated, but I believe it's Tolipocladium. Please let me correct if I am wrong. Clamp connection indicates. So which of the following option generally the clamp connection indicates, like monocharatic mycelium, dicharatic mycelium, both A and B, none of the above. So we know very well that this clamp connection mainly observed in Basidiomycota group of fungi, right? So we can observe in dicharatic mycelium. Again, the special feature of Basidiomycota is the dicharatic mycelium, right? So function of clamp connection to ensure monocharatic condition, dicharatic condition, diploid state, and all the above. So the function of a clamp connection is to ensure the dikaryotic condition of the hyphae. That is the main function of the clamp connection. Meaning of botrytis refers. Botrytis Phytophthora means a plant destroyer. Botrytis probably bunch of grapes. Why bunch of grapes? Because if you see the spores like conidia, are all the spores look like a bunch of grapes or grape bunches. Probably that might be the meaning. Bunch of grapes. Non-parasitic disease of mango. Anthracnose, mango sooty mold, mango red rust, all the above. So mango anthracnose caused by Collotrichum gliosporides, mango rust Sapelioris parasiticus, mango sooty mold, which is a physiological disorder, right? So the answer is mango sooty mold. The conversion of inflorescence into compact masses of sterile flowers in mango is known as. So flowers are converted into sterile masses, right? Sooty mold, black tip of mango, mango malformation. Yes, mango malformation, where the inflorescence they have mentioned, since it is mentioned inflorescence, the inflorescence is converted into compact masses, or I would say the inflorescence is not properly developed, that we can observe in mango malformation. It's a very dangerous disease in mango, but this mango malformation is more severe when it comes to North India only. In South India this is not that much severe, but in North India it is very severe. In banana atrophy symptoms can be observed in which are the following diseases where banana atrophy symptoms can be observed? Bunchy top, cigarette rot, fusary wilt, mocha wilt. First of all, we must understand what is atrophy. Atrophy means where the growth is not proper or the stunted growth. Among the given options, banana bunchy top only where stunted growth can be observed, right? So the answer is banana bunchy top. Spraying 1 hectare of a crop with more than 250 liters of water. So which sprayer can be recommended? They are asking. Low volume, high volume, medium volume. Very for high volume it will ranges from 250 to 400 liters, right? We'll go with the high volume spray. So the answer is probably high volume. I'm not sure, but I believe it's probably high volume. Diseases of flowers was written by. This kind of questions are very difficult because nowadays everyone are writing books, include me. Anyway, jokes apart. So disease of flower crops was written by R. Singh, Mehrotra, Sohi, Muthukumar, and okay. I'm sorry to tell this. This disease of flower crops was actually written by two of our professors, A. Muthukumar and P. Balabhaskar sir. Both are associate professor in Department of Plant Pathology, Annamala University, and they wrote a book called Disease of Flower Crops, which are really fantastic book. Very simple and a very easy book, and that can be recommended for everyone, for students and staff as well. And I forgot where the publishers it is published, but this book was written by two of our professors, A. Muthukumar and P. Balabhaskar sir. Both are associate professors in Department of Plant Pathology, and they had significant contributions, and they have guided a couple of many number of students. In fact, I'm proud to say I'm one of student of both of them. And anyway, the next question is examples of cellulose degrading fungi. Which of the following is an example of cellulose degrading fungi? Neurospora, Cordyceps, Chaetomium, and Pilobolus. Probably Chaetomium is actually degrades the cell wall. Probably Chaetomium globosum. It's actually biocontrol agent, but it is also known to be degrade cellulose, I think. Abnormal bending of leaves in coconut is mainly due to very simple Tatypaka wilt. Okay, which of the following disease is a potato disease that is problematic during storage? They're asking during storage. Soft rot, late blight, potato scab, all the above. Generally is soft rot. The name itself indicates soft rot. Okay, black leg of potato is caused by Streptomyces cabis, Irvine Keratopora subspecies Keratopora, Synchytrum endobioticum, Ralstonia solanacearum, I think scab soft rot. Okay. So all soft rots are caused by Irvine Keratopora subspecies Keratopora. If you see soft rot of carrot, that is also caused by Irvine Keratopora subspecies Keratopora, which produces an enzyme called pectinolytic enzymes, which actually degrades the cells. And of course, cellulytic enzymes produced by Pythium and Phytophthora also can degrade the cell wall. Okay. In which of the disease of cabbage that can produce V-shaped chlorotic rings, it's a very important black rot of cabbage, which is caused by Xanthomonas campestris, Pathophylla campestris. I repeat, black rot of cabbage, Xanthomonas campestris, Pathophylla campestris, which enters into the plant cells mainly through hydathodes, and it is also the example hydathodes, and it is also the example of both the internal and external seed-borne bacteria. It will produce a typical symptoms, V-shaped chlorotic spots. Okay, the sexual fruiting body of a powdery mildew fungus, very simple, Cleistothecium. If you see all the powdery mildews that can produce a sexual fruiting bodies, Cleistothecium. Okay, there are 4 types of sexual fruiting bodies: are there Cleistothecium, Apothecium, Perithecium, Necudaceae. But generally, powdery mildew kind of fungi produces the Cleistothecium kind of sexual fruiting bodies. Okay. So these are all the answers. Please check if I am wrong. For further more information, students can refer this book, A Vision into Plant Pathology, a complete student version. For further doubts, clarification and guidance, students can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.